All right, we're gonna go ahead and get started. Okay, so we're a little bit behind, but that's okay because we've got an extra day built into this whole section. So this is on the cell cycle 2 lecture, and we're a few slides in, and we're still talking about like DNA, some aspects of DNA replication that we're going to need to know later, like when we talk about cancer. Okay, so we're talking here about telomere replication, and it's linked to senescence. Oh, okay, there we go. All right. So, okay, as you all are familiar, there's you need telomeres because there's a problem with the. Let me turn down this volume. Like Snail's not here, right? All right. Well, somehow there's a volume thing. Hold on. Yeah. Okay. Well, it looks like that's what we got. Okay. So, so we need telomeres to protect the ends of the chromosomes. So what's going on? So you've got during replication. If you remember, the DNA polymerase cannot start on DNA. It has to have some kind of template that it's going to add to. So I'll just draw it here. All right, let's say the DNA gets opened up. Oops. Okay, so now you've got a primer that's 5' to 3'. The DNA polymerase can add on to it to go in the 3' direction. But you need to have this primer first. Okay, and because DNA polymerase cannot start on naked DNA, by that I mean it needs a primer. It has to add it to a primer. And because of that, because it can't just, like if it was RNA polymerase has this thing open and it can just immediately start putting down bases. Okay, but DNA polymerase has to add it on to some kind of primer. And since there's no way you can make a DNA primer, because DNA polymerase needs a primer, right? This primer is RNA primer, right? It's going to be put on by Paul alpha primase. You probably learned about this in, I guess, genetics before. So there's always going to be an RNA primer before you start DNA replication. Okay, so if we go back to this diagram, what it's showing you is that at the end of the chromosome, you've always got to start with an RNA primer. Okay, so you got to put RNA primer in. It has to base pair. I mean, the RNA polymerase, Paul alpha primase, the primase part of it is putting in the RNA, and it's using the DNA as a template. Okay, but the cell's can't leave RNA inside a chromosome, so it's going to chew it back. Okay, it's going to get rid of the RNA, and there's no way that you can put an RNA primer out on the end of the chromosome because there's nothing to bind it to. So every time they replicate the DNA, it's going to go in from the 5' end because that's where you put the RNA primer, and then you get rid of the RNA primer. There's no way to put an RNA primer out here because there's no DNA here, right? You can't put an RNA primer here. There's no template. Okay, so every time you replicate DNA, you're going to lose length on the 5' end. Okay, so the way the cell gets around this is by adding telomeres. And with telomeres, you're still going to have the same problem. You're still going to lose length on every time you divide your chromosomes. But the difference is that if this is the end of your chromosome and you're putting in telomeres, and telomeres just sort of a repeated sequence, well, they're going to get shorter and shorter, but it doesn't matter. They don't code for anything, okay? And you'll have thousands or hundreds of base pair dependent species of telomeres. So these are telomere repeats. Okay, so you haven't solved this problem. You're still going to be getting shorter and shorter, but at least you're not losing any genes, which are in the normal DNA. Okay, so the telomeres then are repetitive DNA that just put on one copy after the other on the end of the chromosomes. And these repeats are going to be different for different species. So you can see here the human is A, G, 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 T, T, and it's just that sequence again and again. A, G, 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 T, T, A, G, 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 T, T. Just hundreds of base pairs of this sequence, okay? And it'll be different in every different species. So this sequence is being put on by telomerase, which has two components. It's going to be an RNA that is actually the template for this repeat, and then a reverse transcriptase called TERT for telomerase reverse transcriptase, and so reverse transcriptase just means that it reads off of RNA and makes DNA. So that's a reverse transcriptase. So, and these two form a complex, and that complex is telomerase, and it's the one that's going to add it on. All right, so let's take a look at how it's doing it. So this is the telomerase complex. The green is the TERT protein, the telomerase reverse transcriptase. The blue is the RNA, and they're only showing you the part it's using for as a template. So it's gonna line up on the last two bases, and then it's gonna synthesize onto the 3' end DNA, because it's reverse transcriptase. So it's gonna use RNA as a template, make DNA, and then it's gonna shift over to the next two bases, line itself up again, and again put in another one. Then it's going to shift over again, and it keeps doing that. Okay. So this still, remember, which strand is becoming shorter? Well, it's a 5' strand, right? So you're still going to have the same problem. Even after you do all of this, you're still going to have this strand longer than this strand, because to make this strand, you have to put an RNA primer first, which then gets extended to DNA, and then the RNA primer will get degraded. So there's no way that, so at the end of this process, you're still going to end up with this 3' overhang that you can never fill in, okay, at the end. And nobody really knew how that looked until, oh, actually, this is just saying that telomeres are actually located on the ends of the nucleus, and they're actually tethering the chromosome in place. But, okay. So, but here's my lead in. But nobody really knew what this end looked like, okay, until somebody did electron microscopy of the ends of the chromosomes, and what they see is that it actually forms a loop, okay, the whole thing, the end of the telomere. So this is telomere, telomere, telomere. The whole thing is telomere, and at the end of the chromosome, there's a loop. And so this loop is called a T loop, for telomere loop. And then remember how we have that last 3' extension where you can never fill it in? The truth is that single-strand DNA is a DNA damage signal, and that's single-strand DNA. A DNA end is a DNA damage signal. And so if any of this were exposed, it'd be a DNA damage signal, and the cell would like shut down the cell cycle, you know, going through that P53 pathway that we talked about in the last time. So to prevent that from happening, this 3' end is embedded, is base pairing with itself, okay? Because remember, it's the same repeated sequence. So it's embedded with itself somewhere here. So we're looking at a blow up of this join. So you can see that it's embedded here, and so you don't have the single-strand DNA accessible, you don't have the end accessible, and then this whole thing is coated with proteins, okay? So there's no DNA damage signal, all right? So at the end of the chromosomes, you get these, this one's called a T loop, and then the little one, because it looks sort of like a D, is called a D loop, okay? So it's a D loop, and then the whole thing is a T loop. And it's gonna also, you know, prevent any bad things from happening, like if you were to have DNA, let's say you had these DNA exposed. So what's going to happen? First off, the cell's going to chew this back, okay, to make it blunt. And then non-homologous end joining, which is a repair pathway, is going to take them and then fuse them together. Okay? So you actually lost some sequence because it always chews back to make, for whatever reason, it chews back, makes it blunt, and then even if it was blunt already, it still chews back, and then it joins them. Okay? Non-homologous end joining. So so in a way, you can end up with chromosomes like this chromosome 1 and chromosome 3. They can get merged together, and then you got a problem, right? So these telomeres, by having this loop, are actually preventing a lot of this from happening. Okay. So they're protecting the chromosome ends from nucleases. They're protecting the chromosome ends from fusing with each other through this non-homologous end joining. They connect the chromosomes to the nuclear envelope. That's what I showed you here. So the telomeres are actually linking them on the edge of the nuclear envelope. And, and they're allowing the complete replication of the chromosome, the parts of the chromosome that encode the genes, right? Because you're going to lose some sequence, but it's telomere sequence, not a problem. And like I said, there are telomere binding proteins that coat this whole thing, so none of this is accessible. This whole thing is loaded with proteins that are binding to it. Okay, so there's some interesting aspects about telomeres in mammals. Or probably just in vertebrates, actually. Okay, so in mammals, telomerase, which is the enzyme that's adding the telomeres on, is only produced in eggs, sperm, and adult stem cells. So most of the cells in your body do not have telomerase. Okay, so a lot of your cells are not adult stem cells. Okay, so like let's say you've got a cut, you've got all these fibroblasts which are going to fill in. Well, they're not adult stem cells, and so when they divide to fill in, you don't have any telomerase, and so you're going to have the same problem where the chromosome ends get shorter and shorter. Okay, because there's nothing to extend out the telomeres, and just every time you replicate, you're going to become shorter on that 5' end. Okay, and so on average, if you look at the cells in your body, the telomeres are shorter than when you were born. When you were born, you had longer telomeres, but because most of your cells are not adult stem cells, every time as you've been growing and your cells are dividing, the telomeres keep getting shorter. So you have shorter telomeres than you used to. And then if you take tissue culture cells and you just grow them, you grow normal cells in culture. What will happen is cells will divide and they'll fill up the plate. And then you'll take those cells, you'll take part of them and put them in a new plate. They'll fill up the plate. Then you take part of them, put it in a new plate, and then they'll fill up the plate. We can only do this a certain number of times, and then they'll stop dividing. You'll put them in a new plate, nothing happens. They just sit there, okay? They've gone into what's called senescence, where they're going to not divide, and they're never going to divide again. And during this process, the telomeres are getting shorter and shorter. And you get senescence when they can no longer form that loop, okay? So you need to have a certain amount of space to make that T-loop. If this thing gets too short, it can't actually fold on itself to make that loop, all right? And so what would cause the cells, so I sort of just mentioned it, but what would cause the cells to not divide if the thing was exposed, if that loop end were exposed? Yeah, raise your hand. Yeah, right. So that's a DNA damage signal, and P53 will get activated, and it's gonna express the CDK inhibitor P21, and that's gonna shut down the cell cycle. Okay. So if they divide too much, you get these exposed ends. That's just a non-stop DNA damage signal, and there's nothing that the cell can do about it. Right? It can't form the loop again, and if there's only one chromosome arm, it can't fuse it with something else. So you're going to end up with this constitutive DNA damage signal, and that's going to cause what's called senescence. Okay, so this is this process that I was telling you about. So if you take fibroblasts, which are these like connective tissue type cells around a lot of tissues, you have them in your skin too. So fibroblasts from a newborn human, they're going to divide about 80 times in culture. So if you take one cell, you'll get about 80 cell divisions. So it'll make a lot of cells because it's exponential. but about 80 divisions, and then they senesce. They stop dividing, they senesce. If you do it from a 40-year-old, they're going to divide about 40 times. Fibroblasts from an 80-year-old divide about 30 times. So basically, the older a person is, the less times your cells can divide before they stop and senesce. Okay, and that growth arrest is cell senescence. And there's something that, okay, so like in human cells, these cells will senesce and there's nothing you can do about it. They're just going to sit there and you can look at them all you want and nothing will happen. But in mice cells, there'll be something called a cell crisis, where these cells will senesce and then eventually you'll get some cell growing out. Okay, like, you know, all the cells stop dividing, but then suddenly, oh, something takes over, the population starts growing. Those cells are post this cell crisis, and the cells that grow out are immortal. Okay, they can divide forever now. Okay, so that only happens in mouse cells. It doesn't happen in human cells. Human cells don't happen. Like, there's never been a human cell that just spontaneously starts growing like this. But a mouse cell will, and they'll be immortal. And when they're immortal, they've activated telomerase. Okay, so now they didn't used to have telomerase on, but now they activate telomerase and they can just start growing. So here we're looking at this. This is the age of the human donors in years. And this is how many population doublings there are of fibroblasts. So you can see they start off pretty high, like between 70 and 80 for a newborn. And then there's this rough correlation down. Okay, so this is how many population doublings before they senesce. So you can see that, you know, when you get pretty old, like around 70, you might have around 20 or so population doublings for those fibroblasts. And so this is like an interesting slide which has no real significance for you whatsoever. Just sort of cool and interesting. If you take species that live a long time, so this is the maximum lifespan of that species. So like how long can a mouse live if it doesn't get eaten and doesn't, you know, and it just dies from old age? Or for a human, or a bat, or a rat. So like mice only live about 2 years or so. Rats live a little longer. Got kangaroo rat, got the horse, got man about 100 and some years. That's its maximum lifespan. And then you say, well, how many population doublings do fibroblasts from those species have? And what you see is that the longer the animal is supposed to live, the more population doublings. That means how many times the fibroblasts can divide before they stop dividing. So it's sort of cool. I mean, who knows what this means, right? But, you know, there is a correlation between how much an animal's cells can divide in culture before it stops and how long that animal's supposed to live. Okay, so, all right, so there's a link between telomeres and senescence. Like I said, there's a correlation that telomeres keep getting shorter and then the cell undergoes senescence, all right? And then there's also a correlation that those mouse cells that will grow up and divide forever, and they're immortal, they have activated telomerase, okay? So now this is the key experiment. The human cells, they'll senesce and they'll never grow, right? So now the key experiment is you take those cells and you add telomerase to it, okay? Now the human cells don't stop, they don't undergo senescence, they just keep dividing, okay? So if you add telomerase to human cells, they won't enter senescence at all. And that's definitive evidence that this telomere shortening is what's causing senescence. And as we talked about before, that's due to activating the DNA cell cycle checkpoint. And what's the critical point of this is that it turns out that the telomere RNA is constitutively expressed. But TERT, which is the reverse transcriptase, and if it comes from human, they call it hTERT. That's the one that's only in germ cells, which means like egg or sperm. and adult stem cells. Okay, so it's got restricted. So what they're adding to make these human cells continue to grow forever is they're adding H-TERT. Okay, they're just expressing that one protein because the RNA is already there. The RNA is always there. The H-TERT, you just make the H-TERT, it'll bind the RNA, and then it'll be able to make telomerase. Mm-hmm.
Do you have to periodically add telomerase in culture, or is it just one treatment?
Well, so what they're doing here is they're adding the H-TERT gene, and the H-TERT gene gets integrated into the chromosome. And so there are a lot of cell lines like there's one that we have in our lab called this. You don't need to know this name. IMR90 is a cell line, and it's just a fibroblast, and we get it from the ATCC tissue culture collection, but it only divides like 60 times, 40 to 60 times, and then it stops because it's a primary cell line. So when it becomes immortalized, people usually call it IMR90 H-TERT. So if you ever see H-TERT at the end of a cell line, that means somebody took a primary cell and immortalized it, okay? Otherwise, so these are the only kind of normal cells there are. There are lots of cancer cell lines, but as we'll learn about when we talk about cancer, they're totally messed up. They have like totally different numbers of chromosomes, they're aneuploid, they're really quite messed up. So to have a normal cell with normal numbers of chromosomes, if it's human, you have to add H-TERT, and they'll usually designate it in this way. Okay, so there's certain premature aging syndromes that are genetic. So here's one of them, Werner syndrome. So the gene is a helicase called Werner syndrome, Werner's protein or helicase. So here we see a Japanese American teenager, and here she is when she's 48, and you know, like I'm older than 48. She looks older than me, like a lot older than me. So you know, so she's prematurely aging, and this is a syndrome, okay? And so when I started teaching this class, I was like, whoa, I bet that helicase is required for telomere replication, you know. But I mean, I'm busy. I have other things to study. But eventually, you know, that was the case. It's actually required for telomere replication. You got to open up the DNA and the telomeres. And if you want to look at where their population doublings are, so these are the normal, like, you know, a young person, like a 70-year-old, and they go from like around 70 to 60 down to about 20 or 30. Well, these Werner patient syndromes, you take the fibroblasts out of them, you can see they can hardly divide at all. So they're like 40, between 40 and 50, their population doublings are way lower than just a 70-year-old. So they're much worse than normal. So it's linked again to how many doublings you get before you become senescent. And now there's some new paper which just came out this year. So this is sort of cool. So okay, so you got mice, and then you want to know, well, you know, so as the mice get older, you know, in your body, okay, there's certain, let's say you cut yourself again and again in one place. Well, it's going to be hard to heal that after a while because you're going to have run out of your replicative, your replicative capacity, and they're going to senesce. But this senescence is happening in different parts of your body, right? Like maybe, you know, you're doing something like you're smoking and you're like causing your lung cells to like, you know, keep replicating or whatever. And you're going to be getting senescent cells, okay? So you have senescent cells, and, you know, just from all kinds of things. So they were saying, well, what happens if I get rid of the senescent cells in the old mice, okay? So what they did is, and I'm not going to go into the genetic trick that they used, but they have a gene that's going to be expressed in senescent cells, and then they're going to kill it with this AP compound, but only those cells, and then they... And so what happens is this is the lifespan of a normal mouse. So normal mice live about like, I don't know, like on average like around 600 days or so, and then by 900 days they're almost all dead. Okay, so this is just, you start off with a lot of mice, and every time it goes down, that's like you lost another mouse or 2 or 3 mice. Okay, so this is a lifespan curve. Okay, for a population of mice. And now, if you go in and you just kill, when senescent cells are formed, you just kill them. Okay, so now it's a mouse, everything's the same, but instead of having senescent cells populating their body, you just kill them. Kill them whenever they show up. Now the mice actually live longer. So this is statistically significant. So it's 3 things. It's quite statistically significant different. Okay, so these senescent cells are actually reducing your lifespan in you. And then the really cool thing is, so this was, I think came out in what, I don't have the year here, but it was like last year. Well, this year, the same group come up with a drug that kills the senescent cells. And that makes the mice live longer. So of course they want to test it on humans, you know. So like, you know, what if you killed the senescent cells in you? Would you be like healthier and live longer? So, I mean, it's sort of like, you know, this thing might actually have some applications going forward. All right, so now we're going to shift back to the main cell cycle. We're going to talk about the G2 to M phase transition. Okay, so this was mainly studied in fission yeast, and the reason is because fission yeast, the growth phase is G2, so that's where it spends most of its time, and again, that just makes them really elongated, and then they go into mitosis where they form a septum separating them. Okay, so CDC2 is the CDK1 in fission yeast, all right? And in a wild type situation, they get about this long, and then they divide. But if you knock out CDC2, then because this is the growth phase here, they just stay in G2. They can't enter M phase, and they just get really long. And then you can have a dominant CDC2 mutant where it becomes active earlier than it should be. And now they divide, instead of growing during this phase, they divide too quickly. So they become smaller. All right? And so Paul Nurse, the guy who did the initial screen for CDC mutants in fission yeast, was also studying these other genes that appear to be interacting with the thing, with CDC2 in this transition, G2 to M. Okay? So he found 2 genes, and they had opposite effects. And they seem to be regulating this because you see opposite things when you either overexpress it or get rid of it. And that tells you that it's a regulator of the process. So if he got rid of CDC25, they didn't divide. If he overexpressed CDC25, they divided sooner. If he overexpressed WEE1, they didn't divide. But if he got rid of WEE1, they divided sooner. Okay, so you got things going opposite directions. And it turns out that they're both working on CDK1 and then the mitotic cyclin. Okay, the mitotic cyclin CDK1 complex. CDC25 is activating it, and WEE1 is inhibiting it. So CDC25 activating it, you can see where if you overexpress CDC25, they can divide sooner. If you need CDC25 to activate this and you get rid of CDC25, they won't divide. Same thing with WEE1. If you get rid of WEE1 because it's inhibiting it, they'll divide sooner. Okay? So this is genetically what they're doing with the CDC2. Okay? And what we're going to be looking at is they're going to be both working on the inhibitory phosphorylation sites. So remember, CDK is inhibited by phosphorylation in the ATP binding pocket that prevents ATP from coming in. And that's where they're working. Okay, so this is a diagram, sort of ugly, but it gives the information. So basically what's happening is you're going to have activating phosphorylation and inhibitory phosphorylation. And so WEE1 is the inhibitory kinase that's going to phosphorylate it in the inhibitory site. Now CDC25 is a phosphatase that's going to take that away, okay? So WEE1 is inhibiting the complex by hitting it on an inhibitory site, and CDC25 is going to take it away. Now the CAC, that CDK activating kinase, that's adding the activating phosphorylation, but this is not regulated. It just happens all the time for every CDK. So it's not, as far as we know, it's not regulated. But the inhibitory phosphorylation is regulated here. So WEE1 is going to add it. And then for this thing to become active, it has to be removed. And it's going to be removed by the phosphatase. And then once it becomes removed, you're going to get a positive feedback loop that's going to make them go into mitosis fast. And the way it's going to work is that once this becomes activated, it's going to phosphorylate CDC25 to make it more active, so it removes more inhibitory phosphorylation. And then once it's active, it's going to phosphorylate WEE1 and inhibit it. Okay? So once you make a little bit of this active, it's going to make a lot of it active, because it's going to make it so you get more dephosphorylation of the inhibition to make more of it, and you're going to prevent the inhibitory phosphorylation from beginning. Okay? So again, you want this positive feedback loop to make it a quick transition, not in mitosis, in mitosis. Okay? And that transition is determinant on when the CDK1 becomes activated. And if you mutate these phosphorylation sites in like yeast, now they have something called mitotic catastrophe, where they're not actually ready to go into mitosis, but because they can't be inhibited, they go into mitosis early, and they have a bad mitosis. It's called mitotic catastrophe. And you get the same thing in mammals. There's two sites there. There's threonine 14 and tyrosine 15, and you have to affect both of them. Like you have to make so both of them don't get inhibited, don't get phosphorylated, and then you get mitotic catastrophe. So you can just turn these into like non-phosphorylatable residues. Like instead of threonine, you make it alanine, can't be phosphorylated, or instead of tyrosine, you make it phenylalanine, can't be phosphorylated. And then you won't be able to have this whole thing where you stockpile the mitotic CDK cyclin and then activate it all at once. Now they'll go into mitosis too early. Okay, so we're going to start talking about the phases of mitosis, and then we'll get into some of the regulation of this. So the first stage of mitosis is prophase, and in prophase, the centrioles are separating, and they're starting to reorganize the microtubules into the mitotic spindle. Okay, so here we see them separating, and they're starting to get these microtubules reorganizing. The chromosomes are starting to condense, but the nuclear envelope is still intact in prophase. And then in prometaphase, the nuclear envelope has broken down, the spindles are separated, and they're starting to attach to the kinetochore, which is that part of the chromosome where they attach, and starting to pull them and position them in the center. And so here the red is the DNA, and the green is these microtubules. And then metaphase is relatively quick. This is where the chromosomes are all lined up at the metaphase plate, and it only lasts long enough for them to be lined up. And then in anaphase, they start separating, and you have two anaphases. Anaphase A is where the DNA is separating, and then anaphase B, they're being separated by the spindle poles. The spindle poles separate. Okay? So first the DNA separates, and the spindle poles stay the same, or our centrioles stay the same, and then the centrioles separate. All right, and then telophase is really the rewinding of mitosis. Everything is being reset to go back to interphase. So this is the unwinding of mitosis. The mitotic spindle starts breaking down. The chromosomes, which had condensed, start to decondense. The nuclear envelope starts to reform, and then after that you get cytokinesis. And some people say that cytokinesis is not part of mitosis. They say it's part of M phase, and mitosis is just to here. But for this class, because the whole purpose of mitosis is to make two cells, we're gonna say that cytokinesis is part of mitosis. All right. So I have a movie here, but I think I have to find it. Okay, so I'm going to play the movie. So what we're looking at is a newt. Like a newt is like a salamander, and they have super big chromosomes, and we're going to be looking at it with an Omarsky microscopy. So here we see these chromosomes, and they're being lined up at the metaphase plate. and then they're going to separate, and now they're going to go to telophase. See how they start like decondensing, and they get cytokinesis. Okay, well, let's take a look at this in more detail. Oops. All right. Okay, so we're starting off, and we can see the nuclear envelope's broken down. Let me move this thing out of the way. We can see the nuclear envelope is broken down, and now the chromosomes are in pro-metaphase, where they're being lined up. They're going to be forming a spindle here, but you got a whole mess of chromosomes that are not at the spindle yet. Okay? Now, these guys are being strung along, and they're going to try to end up here at this spindle. Okay? So these guys have to come in too. Now, these guys are in, okay? So now it looks like everybody's at the metaphase, but that's not true. Because this chromosome's not. This chromosome is out of focus, but here it is right here. Okay, now it comes back in focus. And look how everybody's here, but this chromosome's not. They're going to wait for this chromosome to get here before they go from metaphase to anaphase. Follow this chromosome. Comes in, and now when it gets there, boom. See that? Let's follow it again. See, here it comes. Boom. Right there. Okay? So we're going to be looking at that checkpoint later in the lecture today. What is the checkpoint that's keeping the cell from going into anaphase till all the chromosomes are lined up? So then, what you see is that even while they're moving away from each other, mitosis is being unwound. Okay? They're starting in telophase. Look, you got all these chromosomes totally condensed, right? Look, they're starting to melt together. See that? It's like this molten globule. And then you can start to see the nuclear envelope forming around it. And by this point, you know, you've got a nuclear envelope around the DNA, which is decondensed. And then you start cytokinesis. And we'll be looking later at what makes cytokinesis occur after these other stages. Okay. All right, so now, so now we're going to look at some of the major changes in mitosis. Mitosis is a major reorganization of the cell. You got the chromosomes are all condensing. You've got the nuclear envelope is going to break down. There's the whole cytoskeleton has been reorganized. The whole actin cytoskeleton is gone. Microtubules are dissociated, and the microtubules are being reformed into the mitotic spindle. Okay, and then your organelles, they're breaking down too. Okay, so like you've got mitochondria, well, they're fragmenting to become smaller. They're still mitochondria, but they're now smaller pieces. You've got your whole Golgi and ER has turned into little vesicles. And they're little vesicles, so now that they fill up the entire space of the cell, so when the cell divides, you get half of it here and half of it here. You don't have to worry about like what happens to the whole ER and Golgi. It's turned into little vesicles that are just floating around. And the same thing with the nuclear envelope. It's also broken down into little vesicles that are just floating around in the cytoplasm. So a lot of this reorganization is actually caused by phosphorylation by the MPF, mitosis promoting factor, maturation promoting factor, which is the CDK2, or you can call it CDK1, with the mitotic cyclin, cyclin B. So whereas for the G1 to S phase transition in yeast or mammals, we were talking about a single substrate being the critical target. Here, there's multiple things that are being phosphorylated, a ton of things, okay, to set this in motion. And then there's other kinases that are activated below CDK1. CDK1 activates them, and then they do things too, but we're not going to talk about them in class, like aurora kinase or polar-like kinase. They also get activated, and they also phosphorylate things. So there's a ton of phosphorylation that's occurring because there's a lot of reorganization that has to happen during mitosis. And remember, you know, if you just look at a mitotic cell, it's like an interface cell is all flattened out. It's got a cytoskeleton, keep it in shape. And you look at it in vitro, what happens? It just rounds up into a ball, goes into mitosis, and then it flattens out again, okay? So, you know, it's just turned into like nothing but a cell that can only handle going into mitosis, okay, at that moment. And so it loses all connection to other cells also. Okay, so here, as I said, you know, you could think of this MPF mitosis promoting factor, maturation promoting factor, sort of a workhorse. It's going to be phosphorylating lots of things. We're only going to talk about some of them. It's going to be doing chromosome condensation by phosphorylating condensins. We'll just briefly talk about that. It's going to cause nuclear envelope breakdown by phosphorylating lamins, nuclear lamins. We'll talk a little bit about that. Doing a lot of other things, like it's fragmenting the Golgi ER through phosphorylation, particular proteins. We're not going to talk about that. We're not going to talk about how it's reorganizing the whole cytoskeleton and forming the mitotic spindle. So it's doing a ton of things are happening. Like, all those transcription factors that are bound on DNA, all phosphorylated almost, come off DNA. Everything that is normally happening in the cell sort of put on hold during mitosis. All right, so here we're talking about condensins. So the way you measure these things is through a biochemical assay of whether or not it's being active. You would take a plasma DNA and a condensin, and if you had ATP, and then topoisomerase 1. So topoisomerase 1 is just going to release any tension, and and then if you then remove the proteins, what you'll see is you'll get a negative supercoil because the proteins had changed, the DNA had changed its conformation by being in combination with the condensin. And so, that's just an assay that you can use to follow condensin activity. And if you look at that, what you'll see is that interface, the condensins are not that active, and in mitosis, they're much more active in this kind of biochemical assay. It's pretty crude, the assay, though. So just to remind you, what do these condensins look like? So they look like they have two proteins with these cool domains, SMC2 and SMC4, and then they have a cap right here. And the idea is that they're actually condensing DNA by putting DNA strands into this area and capping it. So that they're like bringing DNA together and locking it in place like a clothespin through here. Okay. And then there's condensin 1 and condensin 2, and they differ in their cap proteins. Like here's cap D2, cap D3. We'll talk about cohesins later. They're going to be keeping sister chromatids together, and they look very similar, but they have different SMC proteins. They have SMC 1 and 3, and they have different caps. And then this is the one for mitosis, and then there's a different cohesin for meiosis. It's got a different of this protein that's making the link. All right, but I will tell you that we don't really understand exactly how they're working. Okay, we know that they're condensing DNA. We don't know exactly how they're like looping it together to actually condense it. All right, so now we're going to talk about another substrate of this CDK1, this mitotic cyclin CDK complex, and that's a nuclear lamina. So here you have a nuclear envelope, and below the nuclear envelope is something called the nuclear lamina. And what this is a meshwork of nuclear lamins, and there's like lamin A, B1, B2, and C, and they form head to head and then tail to tail, like this, okay, to make a rigid pattern. Well, it's sort of flexible. That's underneath the nuclear envelope. You actually need to have that to maintain the nucleus as a sphere. Okay, so it's like a rigid, semi-rigid underpinning of the nucleus. Okay, and what happens during mitosis is that they get phosphorylated on their N-terminus and their C-terminus, and I told you the N-terminus links to the N-terminus, and the C-terminus links to the C-terminus to form this kind of pattern. And when you phosphorylate it, so you put on it, so phosphate, is a big negative charge. And when and these things are interacting by charge, like positive, negative interaction. When you put big negative charges on there, it's going to disrupt them. So when they get phosphorylated by MPF or CDK1 cyclin B, the mitotic cyclin, they're gonna fall apart. Okay, so this whole pattern underneath is going to fall apart. And what's going to happen is you're going to end up breaking apart the entire nuclear envelope, and the nuclear lamina breaks apart. Nuclear envelope forms little vesicles, and inside each vesicle is, if you remember the nuclear pore, the center part of the nuclear pore is in the little vesicles, okay? But the basket is gone, and the little tendrils are gone, okay? But you've got the main part that's still like, you know, where it's like the membrane loops in each side, that's still there. So you've got these nuclear envelope vesicles. Some of them have nuclear pore components. Part of the outer and inner part of the nuclear pore is broken down. And that's what it looks like in mitosis. And that's so that they fill up the whole space. So when the cell divides, you get about half in each cell. And then during telophase is where you're rewinding this whole thing. And now these vesicles are going to form on the decondensing DNA. So as DNA stops being condensed and starts like unfolding, these are going to bind on it and then fuse together to remake nuclear envelope. Okay, so this is one of these experiments that shows like how this was determined. How was it determined that in fact the phosphorylation of the nuclear lamina is actually important for the breakdown of the nuclear envelope? Okay, so what they did is they took one of the lamins, A, and it has two MPF phosphorylation sites, two sites that are phosphorylated during mitosis. And they said, okay, well, how important is that, right? All right, so what they did is they took those two sites, which were serines. So serine has an OH group, hydroxyl group, and that's where you would link on the phosphate. But alanine looks like serine, it's another amino acid, but it doesn't have the OH group, so it can't add the phosphate, right? So it's structurally similar, but it can't be phosphorylated. So it's non-phosphorylatable site. So they just mutated lamin A from two serine sites that are get phosphorylated to two alanine sites, and then they said, well, what happens to nuclear envelope breakdown? Okay, so here we have they're expressing wild type lamin A, and here they're expressing this mutant, which lamin A that cannot be phosphorylated. And so this right here is the lamin stain. What you see is that you can see it's sort of high at the edge of the nucleus, and then this is the DNA stain. Okay, and then you see the same pattern if you put in this non-phosphorylatable lamina, because this is before mitosis. Okay, and then here you start to enter prometaphase, and what you see is that in wild type, it's a little hard to see here, but there's a little bit of haze there. That haze is the lamins being dissociated, and you don't see that with the non-phosphorylatable lamin. But the DNA is starting to condense here, and it's starting to condense here. All right. So now, at this point in mitosis, which it's a little hard to tell because, like I said, the whole cell has now rounded up. And in the round up cell, this lamin is filling up the whole space, okay? It looks like it didn't go anywhere, but that's the size of the cell now, okay? And you can see that where the DNA is smaller. And the DNA is condensed, and you're not seeing it, but the mitotic spindle is starting to bind the DNA here. But if you look in this one that can't be phosphorylated, you see that it's still on the outside of the nucleus. All right, so here again, we see that this lamin has filled up the entire space. And here we see that we're in anaphase. The DNA was lined up, now it's separating. And now here, it can't even get at the DNA because the nuclear envelope didn't break down. And now, here you've got two DNA separated. They're starting to decondense. The nuclear lamins are reforming on the DNA, and here you've got a problem where you can't actually have, cytokinesis can't even occur properly because the nucleus never broke down. Okay, so basically what this tells you, the fact is you never broke down the nucleus, and all they did was just express one lamin and could not be phosphorylated. So that tells you that phosphorylation is essential to break down the nucleus. All right, so that's all we're going to talk about right now for getting into mitosis. Now we're going to talk about getting out of mitosis. So to get into mitosis, you had to get rid of this inhibitory phosphorylation. And that makes this CDK1, cyclin B, mitotic cyclin, MPF complex active. And it's doing things, it's phosphorylating things, and you're in mitosis. Okay, to get out of mitosis, you have to inactivate this complex. That complex is why the cell is in mitosis. It's phosphorylating things, making it mitotic. So this complex is driving the cell into mitosis. It's got to be inactivated. The way the cell is going to inactivate it, it's going to degrade cyclin B. Okay? So when it degrades cyclin B, you're just left with CDK1, and CDK1 by itself is not active. It has to be bound to a cyclin. Okay? So we're going to be looking at this degradation of cyclin B, which is essential to get out of mitosis. And in fact, it's essential for the metaphase to anaphase transition. So the metaphase is where the chromosomes are lined up, and then they separate in anaphase. All right. So we had talked about ubiquitin proteolytic pathway before. And like I said, most things in the cell are degraded through this pathway. So is the mitotic cyclin. And they're going to be using a destruction box, degron, which just means that's the sequence that's going to be bound by the ubiquitin protein ligase to recognize it. And that destruction, it's called a destruction box, and it's only 9 amino acids, and it's sort of similar between A, B1, B2. They're all degraded in mitosis, and they need this sequence to get degraded. If you take that sequence and you put it on another protein, that other protein will get degraded, okay? So that sequence is sufficient to make this protein get degraded during mitosis. All right, and as we said, this is going to go through the ubiquitin proteolytic pathway. You're going to add ubiquitin, and that'll cause that protein to get degraded. And it's because there's some ubiquitin protein ligase, the E3, that's going to recognize the substrate, and that E3 is going to determine when it gets degraded, and that E3 is what we're going to focus on. We're going to focus on what's the E3 ubiquitin ligase that's recognizing the destruction box, what's regulating it to allow the cell to get out of mitosis. Okay, so turns out that this E3 is not a single protein. In this case, it's got 8 to 12 components, and it's called the anaphase promoting complex. Okay, because as we'll see, it's required to go from metaphase to anaphase, so it promotes anaphase. So in budding yeast, it's got 12 components. In frogs, only 8 components were identified initially, but now a few more have been added. And so it's a pretty big protein complex. And now we have to get into this Xenopus system that they're using for these experiments. So this is the Xenopus cycling extract. What they're going to do is, so they're using Xenopus because it's really big. Well, I mean, the frogs are about this big, and they've got huge eggs. Their eggs are big, okay? Big eggs. You can see them. They grow them in pools of water in the lab, and when they lay the eggs are on the bottom of the water. So they just scoop up the eggs, okay? And then what they're going to do is they're going to put them in a test tube, and they're in water, right? I mean, if you take them out of water, they're going to dry up. So they're in water, and they're going to add a layer of oil to it. They don't show you the oil on that diagram, but it's essential. So you've got water with Xenopus eggs on top. Okay, you got to have that. You got to have the water because that's where they'll dry up if they're not in water. So okay, so what they're going to do is they're going to start spinning it. So the centrifugal force is going to force the eggs to go. So first off, the oil is heavier than water, but the eggs are heavier than the oil. So what's going to happen is after you spin this, you're going to end up with the water on top, and all the eggs will be at the bottom of the oil. Okay, so what did you do? So by spinning it, you've separated the eggs from the water. And then you just keep spinning it, and you're spinning the ultracentrifuge. So the ultracentrifuge is spinning like 40,000 revolutions per minute, which is about like if I'd say 1001, I mean, it's gone around 400 times. I mean, it's pretty fast, and it's like a huge amount of G-forces. And what it's going to do is it's going to crush the eggs, and then it's going to segregate the eggs into different components. So you're going to end up with the yolk on the top, you get some junk at the bottom, and you have cytoplasm in the center. Okay? So this is undiluted cytoplasm. Why is it undiluted? Because you don't have any water. Water was separated by the oil. Okay? All you did was just take the eggs, crush them, and then keep spinning it till the components separated. Now you got undiluted cytoplasm. Actually, I think I drew this, either one of these people are wrong, me or them. Because they're putting lipid on the top and yolk on the bottom. They're probably right. Doesn't matter, I'm not a Xenopus person. So, but you're separating it, you're spinning it until it's completely segregated and you just have pure undiluted cytoplasm. They're just not showing you the oil here, okay? So then you come in with a syringe and you pull out the cytoplasm. So now you got undiluted cytoplasm. It's exactly what was in the egg. Hasn't been diluted and you haven't taken anything away except for the lipid and the yolk. Okay, so why is it called cycling extract? It's called cycling extract because if you take this, see now you've got this, in a test tube, like this whole thing is cycling extract. All right, I'm going to add DNA to it. What kind of DNA am I going to add? It doesn't really matter. I can add any kind of DNA I want. Maybe because I'm like superstitious, I'll add like sperm DNA to it. I don't have to. I could add plasma DNA. I'm going to add DNA to it. What's going to happen is it's going to form a nucleus. It's going to decondense, and I'm going to get a nucleus. Then it's going to actually replicate that DNA. Then it's actually going to go into mitosis, and it's going to form two other nucleus. And then it's going to replicate that DNA. And then it's going to form two other mitosis. Okay, so it's basically in sync, and it's going through rounds of the cell cycle on its own in the test tube. Okay, so it's going through S phase, and your DNA is going to be replicated, and then it's going through mitosis, and then it's going through S phase again, and then it's going through mitosis. Okay, so it's cycling on its own outside, just in the test tube, with any random piece of DNA that you added to it. Okay, so what they're going to do is, if you remember from Tim Hunt's experiment where he like labeled those sea urchins and say, well, what proteins are being made in the early embryo? We only saw two proteins going up and down. Those are the two cyclins, cyclin A and B, and then ribonucleotide reductase made, but virtually nothing else is made. Because the embryo is loaded with maternal mRNA and protein. And so you don't actually have to make anything. These cleavage divisions will happen by themselves. The only thing you have to make is cyclin. Okay? So what they're going to do is they're going to RNase treat the extract. They get rid of all RNA. Then they're going to inactivate the RNase. And then they're going to add back wild type cyclin B, because that's the only thing they have to make. Just cyclin B, nothing else. Or they're going to add back cyclin B that cannot be degraded. And then the cyclin B that cannot be degraded, that's pretty easy to make. You just take the cyclin B, and you got that destruction box, right? That's a degron that APC, sometimes called APCC, inhibits it, right? It's going to bind it and degrade it. So if you want to make this non-degradable, you just truncate it after that destruction box. Now it can't be degraded, okay? Its degradation symbol's gone. So this is non-degradable. Okay, so they're adding a wild type cyclin B, or they're adding a non-degradable cyclin B that can't be degraded. Okay, so what we're looking at here, this is time, and they're starting their time 0 at metaphase. So this is the DNA, And this is the mitotic spindle because they're staining with microtubule antibodies. So they've got the DNA lined up metaphase plate. At 10 minutes, it's starting to segregate anaphase. At 15 minutes, anaphase is pretty much finished. And then at 40 minutes, they've turned into nuclei. Okay? That's what happens if you have wild type cyclin B. But if you have non-degradable cyclin B, what will happen is, okay, so 0 minutes, your metaphase. At 10 minutes, it looks the same, starting to segregate your DNA. At 15 minutes, anaphase occurred properly. DNA is segregated, same time, everything looks good. But now we're not even at 40 minutes, we're at 80 minutes. And the DNA has not decondensed, and the mitotic spindle is still present to some extent, okay? So what's happening is they can't go into telophase. So they couldn't degrade cyclin B, and they can't go into telophase. So why couldn't they go into telophase? Well, why would you need to degrade cyclin B to go into telophase? So I went over that stage of the cell cycle earlier. So you have to ask yourself, what's happening in telophase? What is telophase? Huh? I can't hear anything unless you raise your hand. What happened in a telophase? Okay, so telophase is unwinding of mitosis. So why do you have to degrade cyclin B to go into telophase? So telophase is the unwinding of mitosis. Why would you have to degrade cyclin B to be able to do it? Huh? Okay, because you're saying the condensins are what? Mm-hmm. Well, yeah, okay, so that could explain that you're not decondensing your DNA. But in fact, it's broader than that, right? The CDK1 cyclin B is phosphorylating everything, or at least starting the cascade of phosphase. It's starting everything. It's breaking down your nuclear envelope. It's breaking down your organelles. It's rearranging your cytoskeleton. All those things are being caused by it phosphorylating so many different substrates. So clearly, you got to get rid of that activity because that activity is driving the cell into mitosis. You can't leave mitosis until it's gone, right? So that's why you can't get through. You can't exit mitosis. So now, this guy Kirshner, Mark Kirshner, he was the one who did that experiment. Now he's asking a different thing. So he knows that APC was the E3, right, ubiquitin ligase, that's degrading cyclin B. And he's saying that you need to degrade cyclin B to exit mitosis. Okay, so that's what he's saying. That's what we just saw, right? So the question is what happens, what is APC required for? Okay, so you got this big APC complex. It's going to be binding cyclin B through the destruction box. And it's going to be adding ubiquitin onto the cyclin B, causing the cyclin B to get degraded. Okay? So now he says, well, what good is APC? What's APC needed for? So what's he going to do? He's going to inhibit APC. And the way he's going to do it is he's going to add a competitive inhibitor of APC. So his competitive inhibitor is just the destruction box peptide by itself. So he's gonna flood his cycline extract with a ton of this peptide. And this peptide is the destruction box. So what's going to happen? The APC is going to bind the destruction box. Now normally it gets rid of the thing it bound because it gets polyubiquitolated and then degraded. Okay? You can't polyubiquitolate this. One, it's going to mainly see this thing. And two, there's no place to add the ubiquitin. So it's just going to bind that, and that's the end of the story. It's inhibited, okay? So he's going to add a ton of this destruction box peptide as a competitive inhibitor. This stuff is going to inhibit APC, and he's going to ask, what do you need APC for? Okay? Clearly you need it to degrade cyclin B, but do you need it for anything else? All right, so this is a little bit confusing because before we had time on the bottom, but this is no longer time. This is how much of the destruction box peptide they're adding. So they're either adding no destruction box peptide, this is what happens normal mitosis, or they're adding increase in amounts. And then here's time. This is all at 15 minutes, and the bottom one is all at 35 minutes. So at 15 minutes, this is 15 minutes after metaphase. DNA is still condensed. It's an anaphase, being separated. Okay? Well, at 35 minutes later, they're in interphase. Okay? They've decondensed. They form nuclei. So now, what happens if he starts adding more peptide? Well, it didn't go from metaphase to anaphase. It stayed in metaphase at 15 minutes. At this concentration is sort of low, 20 microgram per ml. At 35 minutes, you're in telophase. If you add more, you're still stuck at metaphase initially, but now you're at anaphase at 40 minutes. And if you add enough, 60 or 80 microgram, you're inhibiting it completely, and they're just stuck at metaphase. Okay? So what does that tell you? That tells you that you need APC to go from metaphase to anaphase, where the DNA is lined up to go to anaphase. And that's why it's called anaphase promoting complex, because it's promoting anaphase. It promotes it. It goes from metaphase to anaphase. You need it to get into anaphase. Okay. All right. So we'll pick up with this, with what it's actually doing with anaphase next time.